So, in the last module we ended at the discussion on the structure and characteristics of a protein molecule. Let me remind you that in a typical biological system you have large number of protein molecules, and these protein molecules are abundant in any biological cells, and therefore, some understanding that how this protein structure is built up is necessary. So, keeping that in mind, I have discussed that two amino acids, when they react, then they form a peptide bond. So, these peptide bonds has been shown here. So, you have this is that C terminal of one amino acids, C terminal of a protein, and the N terminal of a. So, there is an N terminal of a protein here. So, here somewhere N terminal, and there is also C terminal of a protein, and you have a peptide bond which establish the linkage between two amino acid here, and these peptide bonds are formed, and then this is your polypeptide chain, and in this polypeptide chain depending on the chemistry of R and R prime. For example, this is R prime, the protein molecule have different conformation. And then other things that I have mentioned in the last lecture, in the last module, is that protein has several protein structure can be described at different level. One is the primary structure of a protein, second one is the secondary structure of a protein, third one is tertiary, and fourth one is quaternary. So, each structure, the higher level structure is essentially being built up based on that lower level structure, and as the level increases, the complexity of the protein structure also increases. So, while discussing the different level of structure, I have also mentioned certain length scale that one has to remember. For example, this is a typical helical structure of a protein molecule, and it has a 5 turns. So, total length of this 5 turns is 27 Armstrong, and which each turn is measured to be around 5.4 Armstrong, that is the pitch. So, each bend that whenever each turn this helical structure is being bent, so that angle of bending is somewhere around 26 degree, and this length is around 5.1 Armstrong. So, the interaction between the two protein molecules is also established by very weak van der Waals bonding or hydrogen bonding, which is mentioned in the last module. Also mentioned was that in the quaternary structure can be described either in the wire model or in the space filling model, and this model can be understood by computational modeling of the protein structure, which itself belongs to an entire subject of proteomics or protein structure. So, that goes to the discipline of the biological sciences altogether. The selective binding of a protein to another biological macromolecule is important, and then protein interaction, both are important. So, what I mentioned in the last module is that protein versus biological macromolecule interaction, and also this protein interaction, both are important in the context of particularly biomaterial science, simply because that as I will be dealing in today's model, that cell material interaction essentially mediated by the protein interaction. So, now coming to this cell material interaction, let me explain this central concept, and which is very important in the overall development of these biomaterials, because one has to understand this cell material interaction to the finest level as much as possible. So, let me begin with the typical description of an eukaryotic cell. In the last lecture I have mentioned that eukaryotic cell represents a much more mature development of a cellular architecture with double layer membrane structure. This double layer membrane has a typical thickness of few nanometer. You have a very well defined nucleus structure. In this nucleus you have a DNA, that is a triple DNA has a typical helical structure. Now there are various organelles which are dispersed in the cytoplasm, which include mitochondria and so on. I have also mentioned in the last module that cell membrane has certain characteristics, and these characteristics include presence of certain transmembrane proteins. Transmembrane proteins means, I repeat, proteins which extend from cytoplasm of the cell to the extracellular space. Now these proteins, these integrins essentially take an active part in the cell material interaction. The other things that I have mentioned in the last lecture, just to refresh your mind, that cell membrane also has some well defined pore channels. Now these pore channels have certain specificity towards the transport of the some of the ions. For example, some of the pore channels can allow either sodium potassium exchange to take place between the cell and the cytoplasm and the intracellular extracellular region, whereas some of the channels are specific and they have and they can only allow the transport of the calcium channel, and that too particularly in response of certain external, I mean these are like voltage sensitive. So, the response to the voltage sensitivity, so they allow this calcium transport out or inside the cytoplasm. So, having said this kind of ion transport, particularly calcium as well as sodium potassium, are important as far as the cellular homeostasis is concerned. Particularly calcium is also important when we will talk about this functionality of the bone cells, bone mineralization and so on. Now this is all about cells. Now coming to the proteins, you see the different kind of shapes of these geometric objects I have mentioned here. Now each geometric object essentially represents different protein conformation or different type of proteins. Now these protein molecules, you see this is a solid substrate here. This solid substrate can be either dense, that means non-porous, or solid substrate can be porous also. Porous means it has certain microporosity or macroporosity. The distinction of the microporosity and macroporosity is based on the size of the pore itself. If it is somewhere between 1 to 10 micron, we call typically microporous structure. If it is more than 40 to 100 micron so on, then we call it macroporous structure. Okay. So, whatever is this porosity or dense structure it is, the solid substrate, proteins will get absorbed to this solid substrate immediately after the solid substrate is placed in a culture medium, or in solid substrate is implanted into the animal tissue, then the solid substrate will have immediate interaction with the protein molecules, simply because you have such an abundant pool of the protein molecules. Okay. Now once these proteins gets absorbed, which takes place within just few minutes after placing the solid substrate in a culture medium, then what is the next stage, next level of this one? Next level is essentially is that how these protein molecules, their cell surface integrin proteins or cell surface receptors interact with these proteins which are absorbed on the material substrate. So this interaction essentially takes place like a tongue. You remember a tongue, and this tongue can be used to hold some objects. Similarly, if you consider the transmembrane protein as the tongue, then these absorbed protein as one of the objects that they tend to get hooked to, and therefore, this will be hooked to that individual cell surface receptors. So, this is essentially a physical mechanism which takes place in the early stage of the cell material interaction. Now, other things that I must mention here, ideally a cell has more in a typical freely culture medium or in a culture medium, cell has typically a spherical shape. So, this stability of a spherical object on an otherwise flat substrate is also involved, or it also determines that how a cell would interact with a cell would interact with a material substrate. Now, before going further or before going into more depth on the cell material interaction, let me also tell you that these proteins, depending on the surface weightability characteristics or the surface properties, then protein can get either denatured. So, denaturing means essentially that protein molecules, proteins would not express its desired functionality or would not express its desired function, and then it will not be in a simple coil form, or it is trying to straighten up and so on. So, this example is shown here with the help of this ultramolecular polyethylene, which has typically have a hydrophobic surface. Now, you can do certain surface treatment like oxyplasma treatment or UV treatment or some of the surface treatment to make this ultramolecular polyethylene has a hydrophilic surface. Now, once the surface becomes hydrophilic either by surface or bulk modification, then same protein molecules get same protein molecules are not denatured anymore. So, this denatured and not denatured state are important, because if the proteins are denatured, then it cannot effectively contribute to the interaction with the cell surface receptors. So, it is important for the protein molecules to retain its original conformation, so that it express its desired function. Okay. Now, I mean having explained this one, now let me tell you that what is the next stage or what is the next level of interaction of a biological cell on a material substrate. At the next level, what happens, as I said, that a spherical object, the stability of a spherical object is much more stronger or would be much more stronger if that spherical object now changes its shape, so that it becomes more flat towards the material end or towards the side which gets exposed to the biomaterial substrate. And that is what exactly happens in the next stage of the cell material interaction. You can see that this shape is like off spherical shape. So, this is your off spherical shape, and this off spherical shape essentially allows larger number of cell surface receptors, the way I am drawing in or I am trying to sketch it, large number of cell surface receptors to interact with more adsorbed protein molecules on the biomaterial substrate. So, from the simple description or from the simple schematic illustration, you must be in a point to realize that larger this cell surface receptor protein interaction, more would be the stability of this particular spherical shaped eukaryotic cell. Now, the way I am describing these things, it does not take place so easily or so quickly on a material substrate, simply because any change in the cells, any change in the cell shape has to synchronize with that of the cytoskeletal reorganization process, which I have emphasized to a greater extent in the last two modules. That the cytoskeletal reorganization essentially means that this actin filamentous structure has to be depolymerized and repolymerized. So, this depolymerization, which I have mentioned in the last module also. So, depolymerization means that actin filament is forming lot of monomers, and then repolymerization means these monomers get assembled together to form again actin filaments. And then in the third stage you can see that the cytoskeletal reorganization is also time dependent phenomena, this depolymerization repolymerization. Therefore, the more the time the cell spends in contact with the biomaterial substrates, more is this dynamical cytoskeletal reorganization process takes place. As a result, a single biological cell can be stretched to a maximum extent. And let me also tell you, the more the stretching of the cytoskeleton, or the more the shape change that a cell would experience, more would be the cell adhesion on a biomaterial substrate. Now, the cell adhesion, another term that also is important here is the focal adhesion complexes. Now, these focal adhesion complexes are essentially a cluster of cell surface receptor and absorbed protein clusters. Now, the more the focal adhesion complexes that form on a biomaterial substrate, more stronger would be the cell adhesion on that particular substrate. Now, these points you should keep it in mind, and then we may recall at certain point of time later on during the cell fate processes. Okay. Now, to summarize it, that you have the eukaryotic cells, truly nucleated cells. You also have the cytoskeletal reorganization process, dynamic cell shape changes, focal adhesion complexes formation, and cell adhesion takes place. Now, the moment this cell adhesion takes place on a material substrate, so this cell adhesion, here you have seen that protein-protein interaction, plays a major role, right, which I have mentioned here, that protein-protein interaction takes a major role in the way cell material interaction takes place. So, the moment cell adhesion takes place, next thing is that suppose this is your biomaterial substrate. This entire biomaterial substrate is placed in a culture medium. So, culture medium typically has 10 to the power 4, 10 to the power 5 number of cells. So, it is a large number of cells, and here your eukaryotic cell, here you have this cell surface receptors and absorbed protein molecule interaction, and so on and so forth. So, once this particular cell establishes a rather firm interaction with this biomaterial substrate, what it will do, it will send out the cellular signals or cell signals to the extracellular region. Now, if some of the cells are floating here or some cell is floating here, what it does, they will send the signaling, they will send this signal to the neighboring cells, so that these neighboring cells also would come and also would try to interact with the biomaterial substrate, because this biomaterial substrate does not cause any toxicity to the cell whatsoever. Now, when this guy also comes, this guy also kind of interacts with the same substrates. Now, when you have multiple cells that are lined up on a same substrate, then there is something called cellular crosstalk also takes place. Crosstalk means just like a two human beings, if they stand at a bus stop, so they can talk to each other. So, similarly, that when these two cells, they are sitting on the same biomaterial substrate, they can also establish the crosstalk. Crosstalk essentially means there is a continuous signaling processes that is going on between these two neighboring cells. So, cell adhesion, then cell signaling, and then that more number of cells will come in contact with the biomaterial substrate. Then what? Now, once this cell signaling processes enables that more number of cells to be attached to the biomaterial substrate, the next step would be that we have to understand that whether this. So, let us say if it is a stem cell or if it is some other type of cells, let us say osteoblast cells and so on. So, whether this particular cell when being attached to the biomaterial substrate, whether it would be able to divide the way this particular cell would divide here. So, as I said, the fundamental definition of cell is that cell is a self-contained unit with the capability to propagate itself given the nutrient and proper environment. That means, when you are dispersing the 10 to the power 4, 10 to the power 5 number of cells in a culture medium which contains nutrients, which contains antibiotic, which also contains several protein molecules. The point is that in a freely suspension conditions the cells can grow, cells can divide 1 to 2, 2 to 4 and so on. Now, the point is that whether same cell division process can take place when a biological cell is now adhered to a or now adheres to a biomaterial substrate. So, in other words, so one can see that whether cell division takes place, and that typically people people investigate by culturing them over a different time point in culture in a culture medium. Let me explain this particular point little bit more, and this what I am saying is that suppose you are culturing these cells any particular cell at a given time point. Suppose you are culturing cells at a given time point, let us say this is the point that you are essentially you start the culturing cell you start the cell culture process, and this is your cell numbers. So, suppose these cell numbers if you plot as a function of time of culture here. So, along x axis if you have a time in culture. So, this so, now if you plot along the y axis cell numbers, when you started? Okay, so what I am showing here is that suppose you start the time cell culture at a time is equal to 0, t is equal to 0 time point, then you allow the cell growth to take place for 0, 1 day, 3 day, 5 days. let us consider a hypothetical culture system where you are growing a cell of cell type X for different time period in culture. Now, after the experiments are over, now if you are counting or quantifying the cell numbers and along x axis you are plotting time in culture, let us say in terms of days, then here you started with t is equal to 0, then you are seeing that how cell growth is taking place. So, let us say it is day 1, this is 1, this is 3, this is 5, and then you have a data point which is falling closely more or less like a linear variation. That means, if the cell numbers increases more or less linearly with time, that means cell growth is also linear in nature, and that is a testimony to the fact that cells while being attached to a biomaterial substrate, their cell growth or cell division is not being compromised, or it is like a testimony to the fact this is a kind of a uncompromised cell growth take place. And these things is important for one has to establish, simply because that typically biologists they grow cells in a culture medium without anything, without any substrate on it, or in a culture or in a tissue culture plastic just as a control sample, but not a stiff biomaterial substrate. So, therefore, if you want to describe this particular scientific process to any person who does not know much about the biomaterials, first question they would like to ask you that what about the process or how the cell functionality would be changed, which I will explain to you more in details later on. But at this time, at this stage you must understand and realize that first thing one should check when you are culturing cells on a biomaterial substrate is to see that when you are growing the cells at least 3 different time points and quantify the cell numbers and see whether this cell growth has taken place linearly or there is no change in the cell number. If there is no change in the cell number, which is little bit unrealistic to consider, then you cannot say that this biomaterial substrate, this hatched shaped biomaterial substrate can be used as a cell growth substrate. You can only confirm that point only when this particular kind of behavior you can establish by culturing the cells on the biomaterial substrate. Other points that I must mention at this juncture is that how one can select that what would be the different time points at which you will culture, or what are the different time intervals that you should check that how the cell numbers are increasing or something. So, this depends on what is the cell doubling time. Cell doubling time means what is the typical time frame or what is the typical timeline at which cell is an isolated cell is expected to, an isolated cell in a culture medium is expected to divide. Now, cell doubling time is somehow vary somewhere between 12 hours, 24 hours or 36 hours. This is a typical human cell I am talking about. So, so 12 hours or 24 hours and so on. So, some for example, fibroblast cells, they are fast growing cells. Fibroblast means connective tissue cells, but osteoblast cells, they are relatively slow growing cells, that is the bone forming cells. So, this is the slow growing cells. So, osteoblast cells, they take typically much longer time to divide, and therefore, you may need to extend this just to see that what is the cell growth pattern on a biomaterial substrate, particularly when you use osteoblast cells. You may need to grow them for up to 7 days, because if you grow them up to 1 day, you do not see many of the cells on the substrate, but if you start growing them at day 3, day 5 and day 7, you should be able to see more and more number of cells, simply because osteoblast cells are slow growing cells. However, for fibroblast cells, which is also used in many cell culture experiments involving biomaterials, So, fibroblast cells, since it is fast growing cells, then you can culture them up to 1 day, 2 day and 3 days also. Okay. So, now that cell adhesion takes place and cell growth subsequently takes place. So, cell adhesion then cell growth. The other functionality of the cell, so essentially if the cell growth takes place on a biomaterial substrate, that is a reflection of the fact that cells while being attached to a biomaterial substrate is able to divide. Now, what about the cell functionality changes? Cell functionality changes means, suppose you are growing a stem cell and these stem cells can differentiate. Differentiate means that is the differential cell gene expression. So, can differentiate to other cell types. So, stem cells they can differentiate to, let us take the example of human mesenchymal stem cell or bone marrow derived stem cells. Then it can differentiate to osteoblast cells, for example, bone cells, or it can differentiate to myoblast cells, that is the muscle cells. So, all this cell differentiation process is possible from the stem cells. Now, this cell differentiation means your genes, specific genes related to the osteoblast, specific genes related to the myoblast are to be upregulated or to be overexpressed compared to the some housekeeping genes. Now, the cell functionality changes certainly takes little longer time than the time frame typically you grow cells just to check for the cell growth process. So, this cell differentiation is one of the important aspects, because when you are growing a cell on a biomaterial X or growing a cell in biomaterial Y, one should be curious enough to see that whether that growth of the cell in on a particular biomaterial substrate would also influence the cell functionality changes or not. Now, to answer the questions, you need to grow the cells for a much longer time point, let us say few weeks, let us say 2 to 3 weeks, just to understand or just to confirm whether there is any indication of the cell functionality changes. Now, as I said, that indication many times comes from the way the cell morphological changes. As I said that morphology of the cell is important. So, cell morphological changes essentially indicates any cell functionality changes. Okay. So, the cell morphological changes essentially can be investigated using some of the microscopy techniques. These microscopy techniques are something which is very special to biological sciences discipline and not that much used in traditional material science research. And these microscopy techniques are, one is called fluorescence microscope and another one is confocal microscope. So, if time permits, I will explain to these two microscopy techniques with little bit more details, but at this stage you should know that this fluorescence microscope and confocal microscope, since they are specially biological research oriented microscopes, you need to prepare the samples little bit differently than one normally do in conventional material science research. So, you cannot use the samples just like after culturing you dry and put it on the put it below the fluorescence microscope, put it under the fluorescence microscope, that is simply not possible. You need to do certain sample preparation, you need to use certain fluorescent dyes to excite certain proteins on the certain proteins in the cellular structure before one can record certain fluorescence fluorescence images. So, therefore, not only quantification, but also that how this morphology is changing with time in culture, that also provides lot of important information whether the cell is undergoing any cell functionality changes or no. I think I will stop here and then I will continue this in the next module.